Welcome to another lecture in Imaris Homeschool webinar series. Today we will focus on the Imaris cell module and specifically on the membrane-based segmentation. My name is Alexander Pospiech. I'm Technical and Application Support Specialist and I'm based right in the heart of Europe in Prague. This lecture is following the previous one, which was about multi-component detection in the cell module, about segmentation of nucleus, vesicles, cytoplasm, and then using relevant statistics. One of the most common concepts that is connected to all cell module is right in its name. The cell is more like, it means more like the original name in the very broad, with its all broad spectrum. It does not mean that it has to be animal cell or plant cell or alien cell. It can be anything that resembles this kind of structure. In the cell wizard detection, this lecture starts on this step where we are deciding between the labeling of the cell body or staining of the cell membrane. We have different parameters which are important for the detection. First of them, the source channel. Sometimes it's very easy to jump right to measuring the distances, putting the right numbers, and then we just start the analysis and see, whoa, there is something weird. And maybe in all the excitement about segmentation and detection, we just took the wrong channel. So first thing, pick up the right channel, which is for the membrane. The cell smallest diameter and membrane detail I will explain during the demonstration. Another filter type, smooth and local contrast, again I will explain just in a while. First, how does the cell membrane detection work? We are using a multi-scale watershed algorithm with different options. First, if we have a nucleus, we can use the nucleus as a seed point. But if we have structures or cells without nuclei, we can use this local contrast or smooth options. And then according to that, we will have different sliders and adjustments to segment our sample. We can say that the general rule of thumb is that the local contrast is a default and it also works better for most of the samples. Why would we even have something like smooth then? I will show you right away. With the smooth settings, we are using Gaussian filter that is smoothing the small structures. So for a data set like this, it probably does not make much sense. But if we have a very different or non-homogeneous staining of the membrane, the smoothing can actually help with the detection. On the other hand, the local contrast is actually enhancing the small structures, and it's enhancing the small structures. This filter is also invariant to the intensity of the scaling. That means that it's robust when we have a data set which is bleaching, or for example, if we go deeper to the sample, there is a lower signal, we should still get a good result. So with the local contrast approach. So how do we move from our data set to something where we have our cells nicely segmented? The watershed algorithm or image segmentation, how does it work? We can imagine that the picture is more like a landscape with local peaks, with basins and minimums, and this is how it's cut into the smaller regions. And then there is some automated merge step, which is connecting them back together all the way to the desired result. For local contrast option, we have two sliders with two thresholds, and that is intensity to remove the wrongly detected borders in the background, and quality, which is removing the borders within the cell. For smooth option, we only have the quality option, just to remove the wrongly detected borders. And what is today's lecture going to be about? First, we will look into a rather simple data set, just a 2D image. And we will use some segmentation and color coding according to what is inside the cells. Then we move to complex 3D data set with not so easy visualization and verification. So I will show you some steps. How can you navigate in such a complex sample? And last, I will show you how you can apply the tracking for the segmentation in cell. There was another lecture about tracking, so if you want to know all the details about it, you can again visit our learning section on the Imaris website and see the webinar Become Master of Imaris Tracking. So let's move to the data sets. This is my first image I would like to show you. It's a 2D picture. And let's start right with the cell. For this application, I will use the simplest cell type, and it will be just a membrane without any internal structures. Let's move right away. One small thing, you can always segment just a region of interest. This is very useful when you have a large data set, you are in a hurry, you have slower machine, so you can always try it on something smaller and see whether it makes sense, and then maybe run the big analysis later. Cell detection type. So cell body detection, that was covered in the previous lecture. We will not talk about it. And now we will focus on the cell membrane detection. So first, it is important to select the right channel. Channel 1 is not the membrane staining here. As we can see, these are structures inside of the cell. Maybe right away I can address the question, why don't we use the cell body detection here? First, I want to demonstrate the membrane detection, of course, but also we have cells like this one that do not really have nice staining inside, and we would have a problem with detecting these. And these empty or dead cells, that would not be detected at all. So that's why we are using the cell membrane detection, and with the right channel, which is the red one. And the parameters. Cell smallest diameter, what does it mean? Actually, this is a diameter of the smallest cell. That may be more straightforward wording here. How do I find it out? I can switch to the slice mode and measure the distance. So let's look somewhere at the smaller cells. We have 13 here. It's not exactly straight. Uh, around 12 here. This is not the whole cell. I think generally we are moving around, let's use 12 microns for this one. 12 microns. And then the membrane detail. Again, what does the membrane detail mean? So the membrane detail is more relevant to how smooth or how curved are the borders of the cell. In this case, we can see there is nothing really extreme going on. So the number does not need to be very small, but generally what works very well here is the thickness of the membrane. So if I measure the thinner membrane around 2 microns, this is a parameter which would work well. If you have nicely round shapes like this, the number could be bigger. It should not really cause big problems. But if there is a, like, I don't know, filaments going on, or this is a flat cell attached to the cover slip, it would be better to use the lower number. So in general, the thickness of the membrane works nice. So let's use 2 microns in our case, 2 microns, and we will go with the local contrast method because we have, I would say, very stable staining on the borders in the on the membrane. And here we can see that once we select correct parameters, we can get pretty nice results already. In any case, we have these 2 sliders where we can select the threshold. So first, let's go with the intensity. Uh, yeah, let's put it all the way to the right. And now let's just get the cells we are interested in. Nicely segmented. I think I can go all the way to the settings where it was. Those looked very nice. And the quality, again, let's go to the extreme. You see the watershed segmentation. And now let's remove the borders inside of the cells we don't want. Maybe like this. And let's move forward. And this filter is very important. We are filtering the cells according to the number of the voxels. Why is it important? It would make sense, especially if we have, for example, the cells only in the part of the image and the rest is just a blank space, because that would be detected and labeled as a cell as well. I think the area like this is here. Let's see if, yes, I think this is empty. There are no cells. So let's see if we can get rid of that. That would be, yes, like this, and the second one. So these are not cells, and we just got rid of them. If there were some very small one, that would be the left part of the slider. When you are moving with the sliders in these histograms, you use right mouse button for the right part of the histogram and the left mouse button for this line. Like this, for example. And that's it. We have the sample segmented. I quickly don't even see any mistakes. And let's see what we can visualize here. One very useful function is this slicer and volume display when we want to see more details. What we can now do, because we have these cells which are filled with the green signals and some which are completely empty. So why don't we use the green signal to filter our cells? We can do it first. There is a options really for filtering. So let's visualize the our cells and at this filter but not cell area, we can choose some of the other one. And if we want to sort them or filter them by the green channel, then I would go for cell intensity mean channel 1. So it would be this one, and with this we can filter cells which have good green signal and those which do not have any. We can even create another cells just from these filtered ones. So this would be the original ones. Let's go to volume display. We have all the volume display. Yes, we have all the cells detected here, and in the these filtered ones, we see just those which had the green signal inside. That was filtering, but we can also just color code them. Let's go back to our cells, slicer display so that we can see it nicer. And instead of filtering, we can use the color or statistics coding of these cells. I can check what statistics I have available here, and if I'm missing some of the values, I can just turn them on in the preferences. I will go to Start, Preferences, Statistics, the cell module, and I can see there is dozens and dozens of lines, and whichever statistics I need, I can turn it on or off to hide them from this menu. So in this case, I was use statistics cell area, but I'm interested in the intensity of the channel one of the green channel. And for that, maybe I can use intensity mean channel one. Maybe the spectrum is not the best one, so I can choose any of the other option, for example, this one. And especially when I move to the volume display, I can very easily see which cells are empty and which have the green signal and how much of that green signal is present there. So that was a simple segmentation and some very basic visualization of the data in the 2D image. And we can try uh, another data set. In this case, it will be the complex, much more complex 3D data set. This is from Arabidopsis thaliana. We have only one channel here. And then let's start right away with our segmentation. Again, this is the simple kind of cell with only the membrane staining. There are no structures, no other channels. So let's pick up the easiest type and let's move forward. Again, the membrane staining channel there is only one. Smallest cell diameter, let's measure them again in the slice view. When you have a 3D scan or a time lapse, it's really good to go through the stack and through the time to really look for those smaller ones. Because, for example, here are rather big cells. If I measure those, I can completely exclude anything smaller. So let's see how this one is big. This is only 2.5 microns, 3.5. Okay, I think these are among the smallest. So it's just under 3 microns. Let's use the smallest. We measured 2.5, and the membrane detail again. It can be the thickness of the membrane around 450, perhaps 350 micro nanometers. Filter type will be again the same, local contrast. This data set is a little bit larger, so of course it will take a little bit more time. This is not a specific professional Imaris workstation, it's just a regular gaming computer. Okay, and we do have the first segmentation. Here you can notice the step of removing the largest volume or filtering the cells according to volume will be very important in this one. Now we have the volume display, and well, it kind of looks good, but it may be helpful to move to the slicer display as well. To move the slicer, I can even use the switch between select and navigate here, or I can use the escape button. And it appears that the values we used are reasonably good. So let's just move forward. And now we have to get rid of the empty space, which still this one, which still would be labeled as a cell. So let's filter the biggest one out. Okay. And we finish the segmentation like this. Then we, of course, have the filtering options, all or the statistics that we may need. One of the very useful function, of course, we have overall statistics, but we can look into the selection. For example, I'm interested in this cell, and whatever parameters is cell cytoplasmic volume, for example, I can do it just like that for the selection. If I switch to the navigation mode, I can rotate and select in whatever I need. When I look into detailed, I have list of all the statistics for all the cells. And again, once I highlight some, it will point me into the table. Of course, this data can be exported to Excel and analyzed later. If I need to view help on these settings, I can right click, show help, and it will provide me access to the help file, which is also available for the different statistics parameters and functions. Statistics and show help, similarly cell, and now I can get to formulas and all the details about statistical parameters. Okay, and now let's move to the last sample, and that will be the 3D time lapse. Some of the steps in the cell wizard will be similar. We do have a nuclei staining, so we are choosing this cell type. We will track the cells over time. And now we need to detect the nuclei, so there is a red channel for that. The nucleus diameter, again, we can measure in the slice view, roughly 18 microns. Advanced options are filled also automatically. We can we can go with the default values. Let's make sure we are in the volume display and restrict the value a bit. This looks good. Let's move forward. Uh, Volume of the nucleus, again, I think, yes. Let's get rid of these small particles and go forward. Now cell detection type with the membrane staining. The green channel is for the membrane. Smallest cell diameter, that's again what we measured before. Let's look for those smaller. We can use 20 microns here, and for the membrane detail, 3.5. Let's use 2.5 micrometers. 4, 2.5. Now this part is a bit computational heavy because it's calculating all the membranes in all the time points and in 3D. So this is something that may take a while, especially on weaker machines like mine is now. And we are finished with the big part of the cell wizard. We can filter the cells according to the volume. So we want to get rid of these. And for the tracking of the cells, we will use maximum overlapping with the default value. We also want to track nuclei. This is okay. And for nuclei, we will use the lineage, again with the default values. And let's finish. And now we can simply observe our tracked cells. With the color coding, we can see that now they are differently coded at each time point because we use the object ID. But if we move to the track ID, we can see how the cells are dividing over time. And play the time lapse. We can see how the cells are behaving over time, and we can play our time lapse, observe the tracked nuclei or the cells. Okay, this is everything from today's presentation. I would like to thank you for your attention, and we are ready to answer the questions you may have. And don't forget to join the next week's Imaris Filament Tracer webinar.
Awesome. Thank you so much,
for your attention.
Alexander. So we've had a few questions come in, and I just want to make sure that we cover those. So from Celine, she was asking, can we do image pre-treatment directly in Imaris without having to go somewhere or into another software, but do that before we enter the cell object and go through the segmentation within the cell creation wizard? So can you tell us a little bit about that? And we can, I think you, yep, there you go.
Yes, yeah, absolutely. We can do the pre-treatment of the data sets. There is a image processing option in Imaris. The question was for the Gaussian filter, so of course that's one of the first options, but we have much more functions. For example, the clear view deconvolution is very cool. And if you have the metadata field in the data set, like normally you have from the microscope, it would automatically pop up here. If you don't, you can just fill it in according to how you acquire the data set. So definitely it is possible, and there is a lot of different image processing options here.
Yep, thanks for showing that. Those can also be used within batch processing. So you can set up a batch protocol where you do those, a Gaussian filter or background subtraction or the other options that Alexandra mentioned, and then go into Cell as part of the batch protocol and reproduce that in a number of different images. And you get to walk away and go get a coffee if that's what you want to do. So we do have another one that's come in that's interesting, and it's why would you choose maximum overlap here as the option for tracking? And I think we might answer this together, but you can get started if that's okay.
Oh, you can go ahead.
Okay, so in the example that Alexandra showed, it's that part of it is that we're able to we're looking at these cells, and because we have this edge information, we're able to it gives us better results essentially to be able to track them over time. This is probably not the best answer, but it's the one that we're often choosing when we do have this type of data where we're segmenting them as membranes. And yes, so we've got more information that we're delivering to the tracking algorithm in this case, and so it helps you get better results. Let's see. We have lots of questions coming in at the end. So Anne-Marie is asking, how well does it segment cells of different types or maybe different shapes? And would you need to do 2 different rounds of the segmentation? And so there can certainly be cases where, especially if you're working on like a whole organism or something like that, where you have vastly different cell sizes. In this example right here, there are, I would say these are pretty, there's some big differences here. Thanks for being, doing the show and tell on the other end. And so some you'll have to see, and it depends on your exact situation, how different those cells really, those cell shapes really are. So some of it is trial and error, but also it's totally cool for you to send us a data set and for us to help you work it out and then give you some parameters that work well there. So yeah, definitely reach out to us if you haven't already about that. There's some accommodation that it can make, but there is certainly a limit to it. Okay, let me see. Sorry, I'm scrolling through a list here.
So I would just add that definitely there can be various different types of the cells, and you can maybe run a first run, see whether it works for like a wider spectrum, and then maybe it would be more interesting actually to start sorting them afterwards and export them separately. So for example, in this 2D data set, I have exported separately those which have the green signal. So of course, you can extrapolate it into more various scenarios, and then you can export, for example, 5 different types of cells. So definitely it is possible.
Awesome, thank you. That's a really good point. We also have Carol asking, could the cell module be used for tracking flashes of calcium signal in a cluster of cells? And so we've had a similar question come up when we were talking about tracking. So you might want to go back to that and watch a little bit about it. as long as that membrane label, if you know, in this case, I'm guessing you're meaning, Carol, that you have a membrane label. As long as that membrane label is staying and is different than the label for the calcium signal, they're two different channels, then yes, it should totally work. And then you're going to get a readout of intensity changes over time with that. So, so it definitely should work. Again, to get the specifics on exactly how you would do your particular application, I would ask you to reach out to our sales and support teams that are there taking care of you locally. Okay, so I think for right now, I am going to wrap us up. If your question didn't get answered yet by us in the chat box, Dieter might hang around for a little bit and answer those. We'll also get back in touch with you. Your local team will get in touch with you to make sure that we get data sets if that's what you want to do and that sort of thing. But thank you, everyone. I know everyone's busy with lots of commitments and responsibilities, so we appreciate you coming to this webinar. If you're watching this later, thank you for taking the time to watch it, and thank you to Alexander for giving a great presentation and going over these extra features within Cell that some people don't always use. So yeah, thanks everybody. Take care and have a great day.
Thank you all.